So today we will discuss about the blotting technique. Okay. So what is the blotting? Blotting is a technique for detecting the DNA, RNA or protein sample which is initially present in a complex mixture. Okay. So so suppose there is a mixture of DNA, either DNA, RNA or the protein. Okay, so there is different types of protein, different types of DNA or different types of RNA. So you wanted to identify your target sequence. Okay, so to identifying those particular target sequence, blotting technique is generally used. Okay, and in this that molecules are separated by the gel electrophoresis. Okay, so if it is if you wanted to identify like DNA or RNA, then you have to run the agarose gel electrophoresis, and if you wanted to identify like protein sequences, then you have to do the HDS or the that page. Okay, polyacrylamide gel electrophoresis, that you have to do. Okay, so this gel electrophoresis is a method for separation and analysis of macromolecule, either it can be DNA, RNA or protein, and their fragments based on the size and charge. Okay, so suppose like what is happening in the gel electrophoresis is, if you are doing the gel electrophoresis for DNA and RNA, then this gel electrophoresis is working as a horizontal unit. Okay, and because this DNA and RNA nucleic acid is negative in charge. Okay, so that's why you it will migrate from the negative to positive charge. Okay, it will migrate from cathode to anode. Okay, and based on the molecular size also will play role, like how it is how much length it is. Okay, if it is higher in molecular weight, then it will slowly run, and if it is slow means low in molecular weight, then it will means fastly run. Okay, and likewise the for protein also, like we are giving the means when we prepare the protein sample for gel electrophoresis, that time what we do generally is, in the loading dye there is HDS. Okay, sodium dodecyl sulfate, which is a kind of anionic detergent. Okay, so this anionic detergent, what it will do is, it will give the, like it will neutralize the protein charge and provide the negative charge to protein. Okay, so what will happen, suppose there is a mixture of protein sample. Okay, and each protein is having different charge, right? So that's why to maintain the uniformity, what we generally do is we add the HDS. Okay, so it is a kind of anionic detergent. It will neutralize the the separate proteins charge, and it will provide the anion to these proteins. Okay, so now what is happened is, this all the mixture of protein is having equal charge, right? Now the separation will only occur based on their molecular weight. Okay, means whatever protein is having higher molecular weight, that will migrate slowly, and whatever molecule is having slower means low molecular weight, that protein will migrate fast. Okay, and here there is no means charge biasness will play role. Okay, so that's how you separate different type of macromolecule by using the gel electrophoresis. Okay, then after the blotting, that protein or the nucleic acid sample, you have to transfer onto certain type of blotting sheets, and that after transferring into the blotting sheet, you have to perform any kind for visualization or for detecting, you have to perform either autoradiography or either chemiluminescence. Okay, some kind of chemiluminescence molecule you have to attach with your either probe or antibodies, so that in the visualizing step you can see those macromolecules. Okay, so this is the blotting technique. So there are different type of blotting membranes what we generally use in our lab, that is the nitrocellulose membrane. Okay, then PVDF membrane. Okay, nylon membrane, and DBM and DPT membrane. Okay, so these this nitrocellulose and PVDF membrane is generally used for the protein sample. Okay, and it is also used for the DNA as well as RNA sample, but the most means common thing for using this type of membrane is protein sample. Okay, and this DBM and DPT is generally used for the RNA sample. Okay, so these are the different type of membrane. Apart from this, there are several other type of blotting membrane is present, but this the sensitivity of this type of membrane is very high, so that's why it is very means standard type of membrane which we used in our laboratory. Okay, so then there is a different type of blotting technique. Okay, so there is basically three type of blotting technique. First one is the southern blotting, second one is the northern blotting, and third one is the western blotting. Okay, so in the southern blotting it is generally used to detect the DNA. Okay, so it is identification of specific DNA sequence using a specific DNA probe. Okay, and means in case of southern blotting you are detecting the DNA by using the specific complementary DNA probe. Okay, and in case of the northern blotting it is used for the RNA. Okay, so RNA is the means this northern blotting is used for the identification of a specific RNA using the complementary sequence of RNA probe. Okay, likewise there is western blot is generally used to detect the protein. Okay, so it is here you are detecting one protein by using the specific antibodies. Okay, so this is the different three types of blotting techniques. Okay, that you have to remember. Like southern blotting is generally used for the just a second. Southern blotting is generally used for the DNA identification, northern blotting is used for the RNA identification, and western blotting is used for the detection of protein. Okay, and one more thing here, through using this blotting technique you can only identify that your target sequence, target protein or target nucleic acid is present or not. You can't means localize those protein or those nucleic acid means where it is present you can't identify, because it is the in vitro type of technique, right? Here you are performing this experiment in laboratory. After means extracting the sample you are doing the blotting technique, so you can't identify the live location of those particular protein expression or those particular gene expression, right? So that's why it is just for the if you wanted to identify the molecular weight of those either gene or protein, that you can perform. If you wanted to just identify in means in your sample there is your target DNA or target protein is present or not, the purpose of blotting technique is this. Okay, not to localize those protein or those nucleic acid in the cell. Okay, because this experiment you are performing in the outside of the cell. Okay, so that's why you can't localize, you can't means locate those biomolecule samples. Okay.
Ma'am, one person is trying to join. Can we let him in?
Okay, okay. Oh. But here it is. Okay. Okay.
Thank you, ma'am.
Yeah. So just a second. So this is the three type of blotting techniques: southern blotting, northern blotting, and western blotting. Okay, so southern blotting is generally used to identify the DNA, northern blotting is used to identify the RNA, and western blotting is used to identify the protein. Okay, and another thing is this technique is generally used to just to identify that whether your sequence, whether your protein or whether your nucleic acid sample is present on the mixture of sample or not. Okay, you can't like means localize those biomolecule. Okay, because this thing you are doing outside of the cell, so that's why you can't identify the location of the either protein expression or either gene expression. Okay, so this is to identify just positive or negative, like it is present, whether it is present or absent. Okay, so then we will see like how this southern blotting, northern blotting, western blotting is performed. Okay, so southern blotting, in another term we generally pronounce it as a DNA blotting. Okay, because here your identify the target thing is, target biomolecule is DNA, and you are identifying this DNA molecule by using the DNA probe. Okay, so that's why it is the DNA blotting. Okay, and it is a method which is routinely used in molecular biology detection for a specific DNA sequence in the DNA sample. Okay, and it detect the DNA, means the detecting DNA can be in a single gene, or it can be a part of the larger piece of the DNA. Okay, such as the viral genome. Okay, so that also you can identify, like if suppose this viral genome is this much long. Okay, and so to like which part of the viral genome you wanted to identify, so according to that you have to design the probe, and then it will get complementary to this part of the genome, and it give means the radiography, means here generally if the southern blotting and northern blotting, there is the radiography technique is generally used, because the sensitivity of the radiography is very much high in comparison to the fluorescence and the chemiluminescence. Okay, so but the radiography is very much carcinogenic to the health, so that's why we have to very much carefully do the experiment. Okay, so in the southern blotting, there is, like as I earlier told you, if you are doing the southern blotting or northern blotting, then you have to use the agarose gel electrophoresis. Okay, and in case of the protein, it is polyacrylamide gel electrophoresis. Okay, so for size separation of DNA, and then after that separation you have to transfer the means separated DNA onto a nitrocellulose membrane, and after that you have to hybridize this DNA with the probe. Okay, and then you have to detect it. Okay, so this is the complete procedure of the southern blotting. Okay, so firstly what you have to do is, suppose you are having a complete genome. Okay, so that's why you wanted to only, means you just wanted to know that whether your target sequence is present in this complete genome or not. Okay, so for that what you will do is, you will perform the restriction digestion of this DNA with appropriate restriction enzyme. Okay, so firstly you will chop this DNA into small pieces. Okay, and based on the length, what will happen is it will migrate onto the gel. Okay, so the complex mixture of the fragment is subjected to gel electrophoresis to separate the fragment according to the size, because charge is equal in all the fragments. Okay, so the migration will happen on the base of only the size. Okay, so this will happen, means after gel electrophoresis what you have to do is, the restriction fragment present in the gel are denatured. Now what will happen is, in the gel there is the DNA is double stranded, right? So you have to make it single stranded, then only the probe can come and bind here. Okay, if it is present as a means double stranded, then probe can't bind it. Like in case of PCR, what we are doing is, we are firstly denaturing the DNA, and after denaturation then only primer can come and attach with here. Like that here also we have to denature the DNA. Okay, so the restriction fragment present in the gel are denatured with alkali like NOH or something higher basic reagent. We have to firstly denature it and then transfer onto the nitrocellulose membrane on the nylon membrane by blotting. Okay, so there is a sandwich type of blotting thing is there, like in the lower part there will be one negative charge, because here your DNA is having negative charge, and from negative to positive it have to move, right? So firstly you have to there is a gel, and then in the above this gel there should be blotting membrane, and to do the packaging very tight, you have to add additional sheets on upper side as well as the lower side of this gel. Okay, so then the apparatus will be very tight and there will be no air gaps, so that the electric current will means sorry the in case of the sudden blotting there is capillary action happen. Okay, so that's why the means there will be uniformity in the capillary action. Okay, so if there will be any air gap, then the capillary action will be not very good. Okay, so it can means hamper your transfer process. Okay, so that thing we have to careful look when we are doing this type of blotting technique. Okay, so this is the like how we are means uh, organizing the southern blot apparatus. Okay, so after transfer into the nylon membrane, then what you have to do is the filter is incubated under the hybridization condition, like in the there is a solution. Okay, in a hybridization condition means there is a solution, in solution there is sequence specific probe will be there. Okay, so probe is nothing but it is also a kind of short sequences of nucleotide, which is radio labeled. Okay, or the fluorescence labeled or the chemiluminescence labeled. Okay, so generally we prefer for southern blotting and northern blotting, we generally prefer the radio labeled probes. Okay, so it is either means gamma phosphate labeled. Okay, it is the like radio molecule, radioactive molecule, so that's why we labeled it with the gamma phosphate. Okay, and then when you added this membrane into the hybridization solution, then what will happen is wherever this target sequence it will look by this probe, then it will go and bind to this particular target sequence. Okay, because of the complementarity. Okay, so this process means binding of the probe to the target sequence is called as the hybridization. Okay, because it is forming the hybrid molecule. Okay, so that's why this process is called as the hybridization. Okay, so yeah, the probe hybridized to the complementary DNA restriction fragment. Okay, and then excess probe is washed away by using the washing solution, and the probe bound to the filter is detected by autoradiography, because this probe is attached with the radio labeled molecule. Okay, like the gamma phosphate. Okay, so when the means radiation will like suppose this is the radio labeled molecule. Okay, when the radio light will hit it, this molecule, it will it will emit some certain kind of the signal. Okay, so this signal can be detected by the autoradiography machine. Okay, and this signal will show you like where means these are the result band of your DNA sequence. Okay, this is the target band, you can say. Okay, which is labeled with the radioactive molecule. Okay, so this is the complete procedure of the southern blotting. So firstly what we are doing is we are firstly chopping the DNA into small pieces, because the probe is very much short in size, so that's why to proper binding we have to like perform the restriction digestion, and after restriction digestion you have to run on the gel for the separation of different size DNA. Okay, and after run on the gel you have to transfer the gel into the into the blotting membrane. Okay, and blotting membrane, this is the like organization of how the blotting how the gel is transferred to the blotting membrane. okay. And then you have to this after transferring you have to keep this membrane onto the hybridization solution where the probe is present, okay. And this probe will go and bind to the complementary sequence of the means restrictly digested DNA sequence, okay. And the extra probe which is not bind with the this sequence, it will be washed out using the washing solution. And then this probe will be detected by the autoradiography machine, okay. And because of the means radiation emission, okay. So which reveal the DNA fragment to which the probe hybridized, okay. So where the probe will bind, it will emit certain kind of light, okay. And that light will means those particular places the light will be shown. So from here you can find out that in which sample you are having the your target sequence, okay. So this is the southern blotting. Then the another technique is the northern blotting, okay. So it is called as the RNA DNA blotting, okay. Because here you are sorry, it should be yeah, it can either be DNA or it can either be RNA also, okay. So the probe can be DNA as well as the RNA, okay. It's depend on you what you wanted, okay. So but the target sequence will be RNA, and the probe either can be DNA or it can be RNA, okay. So here in case of the northern blotting, you no need to perform the restriction digestion of RNA, okay. And this because it is already single standard, so that's why here there is no need to denature also, okay. Because it is already single standard, okay. So use for and the gel is make of formaldehyde to break the hydrogen bond and denature RNA, because single standard RNA will not intermolecular base pair and fold on itself, okay. So like to break the hydrogen bond, we are generally using the formaldehyde in case of when we are running the RNA onto the gel. So that in that gel we are we mix the formaldehyde so that it can't be form the intramolecular hydrogen bond, okay. And this is single standard, so there is no nature to means no no need to do the denaturation, okay. So apart from that, the northern blotting is means almost similar to like the southern blotting, okay. So here the RNA is isolated from the several biological samples, okay. And after isolating it, what you have to do is samples are loaded onto the gel and the RNA sample are separated according to their size on the agarose gel, okay. And after means size on the agarose means after separating on the agarose gel based on the size, then what you have to do is you have to transfer the gel into the blot, like in case of the southern blotting what we did, okay. So this blotting is like the most common blotting is the nylon membrane or amino benzylo oxymethyl filter, okay. So by creating sandwich kind of arrangement, you can transfer the gel to blot, okay. So after transfer into the blot, then the membrane is placed into a dish which is containing the hybridization buffer or hybridization solution where the labeled probe is there, okay. So this labeled probe will go and bind to its complementary region of the sequence, okay. And it after means during the autoradiography, it will show some certain kind of the band wherever the probe will hybridize, it will show the band, okay. So this is the autoradiography picture, okay. Wherever the probe is hybridized, that is showing certain type of band, okay. That is detected by the autoradiography machine, okay. If it is labeled with the radioactive means molecule, then it is called as a autoradiography. And if it is labeled with any kind of chemiluminescence reaction, so it will be it is told as a chemiluminescence machine, okay. So in both the cases you can observe the band, but the autoradiography is much more sensitive comparative to the chemiluminescence, okay. So this is the Nathan blotting. The overall procedure is same in both the cases. Just the initial procedure is little bit different. Like in case of the DNA, you have to, like, it is a very big genome, so that's why you have to digest into small pieces, and then again, because it is a double standard, so you have to, like, do the restriction digest, sorry, you have to denature it. Okay, but in case of the RNA, it is a small molecule, so there is no need to, like, double means restriction digestion as well as the denaturation, because it is already single standard. Okay, just you have to like run on the formaldehyde gel electrophoresis, so that the intramolecular hydrogen bond can't be formed. Okay, otherwise what will happen, this is suppose RNA molecule, so this hydrogen bond will bind with each other, okay, and form the intramolecular hydrogen bond. Okay, so that thing is only different, otherwise the overall procedure is same in both the cases, the southern blot and the northern blot. Okay, then the next one is the western blotting. Okay, so the western blotting is also known as the immunoblotting or protein blotting. Okay, it is a technique used to detect the presence of a specific protein in a complex protein mixture according to their size and amount. Okay, and this other word, this western blot is used to determine the protein expression. Okay, it is a core technique in cell biology, molecular biology, and virology and others. Okay, so unlike the southern blotting and northern blotting, here what is happening, here you are not using any kind of probe to detect the protein. Okay, here you are using another protein which is a antibody. Okay, which is called antibody. Here you can see, suppose this is your target proteins. Okay, so you have to add, to detect this protein, you have to add antibody instead of the probe. Okay, this antibody, it can either directly linked with any kind of the chemiluminescence material or any kind of the radioactive molecule, and or you can use this antibody as a primary antibody which is against of this particular antigen, and then you add secondary antibody which is complementary to this primary antibody, and this secondary antibody is labeled with certain kind of the enzyme, and when you add the substrate to this whole solution, this enzyme will act on the substrate and it will convert it into the product. That product you have to detected by the chemiluminescence or the fluorescence. Okay, so this is the western blotting. Okay, so this is the complete procedure. So firstly what you have to do is, you have to if you wanted to, like, isolate the protein from the cell, so you have to firstly, like, lysis, do the lysis of either tissue, either cells. Okay, so firstly lyse the cell so that the protein will come out. Okay, and then you have to these are the protein extraction method, either boiling, sonication, homogenization, and motor grinding through liquid nitrogen, we can isolate the protein. Okay, so once the protein will obtain it, then we have to perform the SDS page. Okay, so sodium dodecyl sulfate polyacrylamide gel electrophoresis. Okay, so here what is happening is, the protein is having, some will be having negative charge, some will be having neutral in charge, some will be having positive charge. Okay, so that's why there is no uniformity. So to means and this polyacrylamide gel is only moving from negative to positive charge. Okay, so that's why what you have to do is, you have to add the SDS, which is the anionic detergent. Okay, so this anionic detergent will suppose this is the protein molecule, so it will provide the uniformity to the mixture of protein. Okay, by giving the negative charge. Okay, so it is an anionic detergent, so it will give the anion to the protein mixture. Okay, so then now the every protein is having the same charge. Okay, so now when you run on this polyacrylamide gel, then based on the molecular weight it will migrate. Okay, not on the basis of charge. Okay, so it will migrate based on the their molecular weight. Okay, suppose the molecular weight is higher, then it will slowly migrate. If the molecular weight is very low, then it will migrate fast. Okay, so after once the polyacrylamide gel electrophoresis, you have to transfer this gel into the blotting membrane. Okay, here you can see the protein you have to transfer. So protein is now in the gel. Okay, so you have to transfer this protein from gel to onto the blot. Okay, like this. Okay, so suppose this is the HDH gel, here the protein is there, so you have to transfer onto the membrane. Okay, so there is the equipment of western blotting. Okay, so here you have to, like, again you have to make a sandwich kind of thing. Here you can see the cassette is there, then there will be some sponge. Okay, and then your gel will be there, and then here your membrane will be there, then again your sponge, and the negative and positive plate will be there, and you have to tightly pack it and then run on the means under the current. Okay, so then what will happen is from negative to positive it will migrate. Okay, and it will come to the gel to membrane. Okay, so after transfer into the membrane, then you have to perform the blocking. Okay, so blocking why we do is in the membrane there is different spaces where can the antibody can non specifically bind. Okay, so to means inhibit those kind of binding, we perform the blocking. Okay, so after blocking we have to add the primary antibody which will be specific to our antigen. Okay, and after adding the primary antibody, then we have to do the washing so that the extra antibody can wash away, and then add the secondary antibody which will be either horseradish peroxidase labeled. Horseradish peroxidase is a kind of enzyme. Okay, so this labeled secondary antibody will bind. Okay, here you can see that primary antibody firstly you have to bind, which is specific to this antigen. Okay, and then secondary antibody you have to add, which is specific to this primary antibody, as well as this is having the HRP labeled. Okay, HRP is a horseradish peroxidase. It can be other enzyme also. Okay, so the most common one is the horseradish peroxidase. Okay, so it is labeled, and when this secondary antibody is bound to the primary antibody, now what you have to do is for detection, like to see as a band, what you have to do is you have to add the substrate. Okay, so wherever the enzyme is linked to this primary antibody, there the enzyme will convert the substrate into product, and on that place it is showing the band. Okay, which is called means as a your target band that you can develop. Means the visualization you can see by the X-ray film, or either luminescence detection and either fluorescence detection. So it is all depend on you, like what kind of conjugation is there with the antibody. Okay, it can be radio labeled, it can be chemiluminescence molecule, it can be fluorescence labeled. Okay, so this is the complete procedure of the western blot. Okay, so in case of the western blot, there is no probe is using, instead of probe there is the use of antibodies. Okay, so yeah, so this is the western blot. Okay, so there is a comparison of southern, northern and western blot technique. Okay, so in case of southern blotting it is the DNA molecule, in case of northern blotting it is the mRNA molecule, in case of the western blotting it is the protein molecule. Okay, and the gel electrophoresis is agarose gel. Okay, in case of northern blotting it is the formaldehyde agarose gel, and in case of the western blotting it is the polyacrylamide gel. Okay, and gel pre-treatment means before running on the gel you have to do the depurination, denaturation and neutralization. Okay, but in case of northern blotting there is no need of denaturation and digestion. Okay, and western blot also there is no need. Okay, and the blotting technique means the blotting method or the transfer method generally happen in case of southern blotting and northern blotting by capillary transfer. Okay, so just you have to means keep your membrane onto a solution, so that capillary through capillary action it will migrate from lower part to upper part. Okay, but in case of western blotting you have to perform the electric transfer. Okay, so there is mean the transfer happen in the presence of electric current. Okay, and this probe is DNA, which can be radioactive or non-radioactive. Okay, and in case of Nathan blotting, it can be complementary DNA, it can be complementary RNA also. Okay, so here, like in case of Nathan blotting, you can use DNA and RNA as a probe. Okay, and it can be also a radioactive or non-radioactive. In case of the Western blotting, it should be primary, means antibody, not the probe. Okay, and this antibody can be also radioactive, non-radioactive. Okay, and the detection system here, autoradiography, chemiluminescence, colorimetric. Okay, here also autoradiography, chemiluminescence, colorimetric. Here the most common, in case of the Western blotting, we generally don't prefer the autoradiography. Okay, we generally prefer chemiluminescent and colorimetric. Okay, so these are the differences. Apart from this three blotting technique, there is, in now recently in a days, Eastern blotting is a kind of blotting technique. Okay, which is most commonly used for the protein post translation modification. Okay, so it is the, we can say like it is the extension of the Western blotting. Okay, because here it is also identifying the protein. Okay, not the protein expression, but how it is, means what kind of post translation modification is there, it is detected by Eastern blotting. Okay, so suppose there is a lipid molecule is attached to the protein, if suppose there is a carbohydrate attached to it, so this type of detection you can identify by the Eastern blotting. Okay, so it is the extension of the Western blotting. Question is, which one of the following technique is used to monitor RNA transcript both temporally and spatially? What will be the correct answer? No, Nathan blotting will be not the correct answer, because as I earlier mentioned you that this blotting technique is not for the real time identification or real time location. Okay, temporally and spatially means when you are comparing the your location or something, means something, some parameter, when you are considering with the time. Okay, so that is called as the temporally and spatially. Okay, with the time when you are comparing something, then it is not, means blotting technique can't do that thing. Okay, so this is only just like whether your target thing is present or absent in the complex mixture. Okay, it can't tell you like where it is present in inside of the cell. Okay, so that can be identified by the in situ hybridization. Okay, so the correct answer of this question will be in situ hybridization. This is nothing but the same thing, but here what you are doing is the whole process, instead of doing outside of the cell, you are doing inside of the cell. Okay, so the probe you have to firstly transfect inside of the cell, then this probe will go and bind where the nucleic acid will present, and go and bind with the because of the complementarity. Okay, and it will give some kind of the signal. Okay, that signal you will identify by the either autoradiography or either by chemiluminescence, fluorescence, and you can tell by looking onto the position where this fluorescence is coming. You can tell like exactly on a particular time period where it is. Okay, so this thing can be means done by the in situ hybridization, and through in situ hybridization you can only identify the nucleic acid sample, not the protein sample. When you are talking about the protein sample, then you have to perform the immunohistochemistry or immunofluorescence. Okay, there like the procedure will be again same, but there will be like antibody will go inside of your body, inside of your cell. Okay, so this is the difference. When it is talking about both temporally and spatially, it means it is talking with respect to the time. Okay, means at a present time where it is. Okay, so that's why the correct answer of this question will be in situ hybridization. Okay, got it?